It's a lot of data. And then what we use is statistics to test for association, to look for patterns. Okay? The human eye is probably the best way to look for patterns. So if you look around, you know, out into the fields or something like that, your eye quickly distinguishes patterns. You know, we can find, you know, groups of plants that are flowering or butterflies. So we are very good at that. But when we look numbers, we have to develop statistics to be able to help us to find patterns, which are much harder to see. And I will tell you about how this is done. Okay? So the methodology, if it is for a human population, it looks like this. You have a group of individuals that they are healthy, here a group of individuals that they are not healthy, then you get all the markers for all the genome, and then you are looking for a statistical value that it tells you all these individuals that have the disease have these markers, okay, in the same part of the chromosome. And this tells you that with some confidence that there is a gene in that location that controls the disease. And if you have 3 peaks, that means that there are 3 genes, right? This is for humans. In humans and animals, you cannot control genetics. But if you work in animals that we breed, like cattle or chicken, then what you can, and plants, what you can do is you can engineer populations different to the way that we do human populations. And you can do it by taking 2 parents that they are different, making an F1 hybrid, and then from here make an F2 population. And this population, it has different phenotypes, okay? And like in the previous case, you make a group that is one phenotype, the other phenotype, and then you look for markers associated to these phenotypes. It's the same, but it's built in a different way, okay? And it's a bit more clever, so allow us to use the markers in a better way. Okay, there are some considerations to this technique. It's not as simple as it looks. The first thing is the quality of the data. So you need to have, you know, a very good confidence value that the phenotypes that you look at are true and that the genetics are true. So you cannot have margins of error. And then genetically, there are some limitations. If you're looking for humans, then you are looking for confounding effects, like could be sex, age, or health, correlation with other variables. But another important thing, 2 important things, is strictly genetics, which one of them is called population stratification, and the other one is called linkage disequilibrium. Don't worry about this, I'll explain in a minute what it means, right? This is quite a problem in these studies. So the population stratification, what it means is that there is a structure in the population of the individuals that you sample. So if we were doing a GWAS study with only you in the class, then you are genetically relatively similar because you come from Indonesia. And that means that you will be in groups, right? And then maybe we can find somebody from Java here in this group, somebody from East Timor here. So you can make actually groups. So your genetics is already determined from where you come from, and this is not good, because that means that all these individuals are the same and the same. So the number of informative markers that we use is very low. And that is a problem in humans and other studies. So this is because there are differences in the frequency of different populations, and the statistics that I told you for GWAS assume that there is no preference for any particular population structure in the sample that you are taking. Okay? This creates what we call confounding effects in populations, and this is something that is a big problem in humans for medical studies and GWAS. Because if you look at these populations and you calculate GWAS, if you have a lot of individuals that come from these, let's say from Java, then you will assume that the results you get are true, but they are not true. They are only associated probably with these individuals and not with the others. So there are solutions to this. The first solution is to visualize that if you have a sample, you have stratification in the populations. There are two strategies to do this. One of them is phylogenetics. So you take samples, I can take samples for all of you, run 10 markers, and say, okay, are you different or not? And then a phylogenetic tree will tell us very quickly, how many groups we have in this class. Okay? The other way is to use what we call a principal component analysis, which what it means is taking all the data, throwing all the data all together, and statistics will make groups according to the similarity between them. And you have a big group that will share the same markers, then it will look like this, and all the others will be scattered all over the different axes. Okay? It is possible to correct the data, even if you have that problem. One of them is genomic, so it's basically selecting individuals that don't have the same genetic makeup. And then this allows you to create a new principal component analysis that allows the data to be scattered everywhere. So then you can actually select individuals that they are not grouped together, but they are more scattered all over the place with the data. So that is one problem. The second problem, which is in a way related, is called what's known as linkage disequilibrium. Okay? And this what it means is that the alleles that we find, the different variants, are not independent from each other. So what it means is that if we have different variation in one chromosome, then all of us, we share the same information in that part of the chromosome. So maybe this class is very different at the genomic level, but because you come from Indonesia, maybe you have been selected to like durian, let's say, right? And because you like durian, you have selected a part of your genome with some genes that allow you to taste durian like a nice thing. And what it means is that this creates a lot of parts of the genome which are conserved with all of you, and the rest is different. Okay, I'll just give you an example. And by the way, I don't think there is a gene for durian. Maybe it is. Okay, so the problem is that you have too much linkage disequilibrium, then it will look like that. If you look at polymorphisms, they all look very uniform. So then you don't have any differences in that part of the genome. Okay? But sometimes these kind of groups could be interesting. Like for instance, if the durian-like gene in the Indonesian population is clustered in this region, this is very good because then we know that this is where the gene that is important for like durian is located. Okay? So it's not good for genome-wide association studies, but it's good to identify that gene that is connected to the phenotype. But this is rare. It's not quite common. So in the standard with GWAS uses methods that to be able to overcome these limitations. One of them is the univariate method. Okay, that's the standard GWAS. But if you want to correct the data, there are at least two methods that we use. One is called the multivariate, and this one penalized for regression methods, factorial methods, and these two are very well established statistical routes. I don't have to tell you what they are, it's not important. But if you have a study where you are doing GWAS, then you can just upload in your computer this kind of software, and you impute the data, and then the statistics will do the calculations for you. It's not, you don't need to know this for now. You really want to know more about it. So I'll tell you more about this one and a little bit about that. So if it is a univariate method, what it happens is that you have a matrix where the data will look like this. You have a group of individuals that they don't have the disease, a group of individuals that have the disease. Then red and blue is the different variants of the different genes all over the genome. So the matrix is huge, right? And then you are trying to look for polymorphisms that they are correlated. And then you have a patch group of information for all the individuals. So this is a huge matrix, right? So this method, what it does is group nucleotide polymorphisms one by one and compare them, test them to the phenotype. one by one. So you use statistics for test and use a correction for multiple testing. For testing, this will become pretty familiar to you. So one of them is Fisher exact test. This is a very standard statistical test for biologists. Okay? And the other one is a gold standard, that is the Fisher exact test. So this is the more advanced test. So yet again, you don't really need to know how the Fisher test works, but as far as you know the question and you can input the data there. In fact, there are programs that they use this univariate method and it will calculate everything for you. But you need to know that there is a correction with the statistics. And the correction for Fisher test is called Bob-Ferroni. That is the standard correction. And then the standard for determining how robust your findings are is what we call FDR, which stands for false discovery rate. So this basically tells you if the results you are getting, you can find it just by accident, just by random. It tells you the how. So there are strengths and weaknesses of this. The strength is that it's a very straightforward. Okay. Well, of course, you run in your computer, so it's very straightforward, right? But as a method, you know, for development, it's straightforward. The computational analysis is very fast, so it can be done literally in a few minutes in your computer. It's conservative and it's easy to interpret. So this is the plus. The weaknesses is that it's not as strong as the multivariate method. It has effects due to the size of the individuals or the number of SNPs, so you don't have a big matrix, it's not as robust. What you have to think is that when you do these studies, you have to sequence genomes or have genotyping information, so this costs money. And that means that for many studies, you cannot spend a lot of money. So there's a limit on that. There are some effects of some SNPs that maybe will send it to the wrong location because they're very strong. So this could be a problem. So another strategy is to use the multivariate method. Okay. Oh, sorry, before I finish that. You use regression models, shrink the data, and use two methods, which is one of them is lasso, which is the gold standard, which is the most common statistical method that people use for correcting the data. Okay. So lasso is a method that uses a linear model, and this is how linear models usually look like. So you have different variants, and then variation from zero. So you can have this. What it means is that if you are looking at the frequency of polymorphism, it could be 1 or 0, but when you look at the data, it's never like that. So you get values of 0 point something that it doesn't give you information. So these methods, what they do is adjust for this variation in the data. They normalize the data so they become either 1 or 0, and they use different coefficients to be able to correct from the data. It's not important for you to know that, all the details, but just you need to know that there is a correction method that is called lasso, which is widely used for that. I will tell you later why this is important. Okay? So the strength of this is that it's a very stable method when you have correction. It's different to interpret, easy to interpret, sorry, and then likely is accurate to select for multiple predictors. The weaknesses is that it has multiple linearities, and it's not designed for things that they have high p-value. So high p-value are data that they are below the statistical cutoff, okay? Because it's a very stringent method, and it's computationally very intensive. Because like in the method that I told you earlier, use the matrix, calculate all the values, make the correction in all the values, adjust the values again, calculate again. So it's a really long, lengthy process. Okay, it could take several days. But, okay, once you take the data, put it in your computer, select the method you want to use, believe me, you will know the statistics. Then what you get is this, okay? And this is what's called the Manhattan plot. And a Manhattan plot, what represents here, is a way to display an association between the phenotype and the genotype. For you to understand this, this is human data. So you have in every block here different colors, it's a different chromosome. With dots represents the frequency of nucleotide polymorphism in the genome, in the population, in that matrix, okay? And the association between that marker and the phenotype. Let's say this is durian taste, right? So then you compare, okay, how many people say they love durian? Then you look at the genetic information in the matrix. Then you calculate the statistics. And if it is somebody that it doesn't like the durian, most likely it's not going to be put into the positive class. But if you have said, I like durian, you will be considered strongly. And if you get many people that they share the same marker, will have a cutoff, okay, for high confidence statistics. And what you can see here is that, well, you know, there are many passed genes in the chromosome that they don't tell you anything about your taste for durian. But there is one here that is very, very high, okay? And what it tells you is that probably this is a group of 50 or 100 or 200 individuals that said, I love durian, and they have the same genetic makeup in one part of the chromosome. And this is telling you statistically there is a gene there that controls that phenotype. Okay? And this is not a very clear GWAS. I'll show you later another GWAS. This could be another gene that you can see here that it might be associated with the phenotype because it's right on the cutoff and it has many different markers in the same region that they are connected. And this is usually a very positive sign because when genetics selects for a variant, it doesn't select only one position. Usually you select a lot of different markers nearby because as humans, we are hybrids, right? Our mother and our father were different, and then they all have different markers, and then we always have a combination of both. Okay, so if we think about what these kind of studies have told us about humans, this here is the 22 chromosomes of humans, and with these circles represents all the genes that they have been associated with disease in humans by GWAS studies. It's a lot. In fact, we have probably explained almost 50% of the genes associated with disease in humans, maybe. And this was in 2011, okay? So there are around 1,500 published GWAS studies, and there are around 250 phenotypes in humans that we have. What do we know from these studies? Okay, so the first thing is that, which is a surprising finding. The first one is that not all the things that explain the phenotype are in the gene. And this is strange because as a geneticist, and probably you are biologists, you think about a phenotype, you think about a mutant. When you think about mutant, you think about a gene that makes a protein. But we humans don't have too many mutations in the genes that gives phenotypes. And this is why the phenotypes we have are not always in genes. So very few are in genes, and most of them are in regulatory elements. So, and I will tell you exactly why this in a minute. Okay, and if they are in regulatory elements, means that they are in regions of the genomes that they don't have genes. And I can tell you another thing connected to phenotype. So you are from Indonesia, share a very similar genome, but if you look at you, you see that you are very different in, you know, color of your skin, eyes, the shape of your eyes, And this is not exclusively related to the genes you have, but it's related to these regulatory sequences outside the genes that control the way that we are. And we are all like this, right? So the important regulatory elements are not in genes, but they are outside genes. Okay? So when we do this kind of studies in humans and we find a strong GWAS, then this looks a bit better, Manhattan blot. So these are for different diseases. So this is coronary artery disease. You can see it's a very strong GWAS signal compared to the data across the genome. In some diseases, like Crohn's disease, this is a intestinal disease. This, at least, there are 4 genes. You can see 1, 2, 3, and 4 genes, or groups of genes. Sometimes you see 2 genes. But the interesting thing is that when you can look at some diseases, what you find is that some genes don't have anything. And then you have to think, well, why do not have anything? Well, one possibility is because we cannot detect it. And the other possibility is that these diseases are not genetic. So if they are not genetic, they are not, we cannot detect. And this is bipolar disorder. This is a behavioral disease. This is hypertension. So these tell us that either there are many, many genes, a huge amount that we cannot detect, or that they are not genetic at all and they are purely environmental, or 90% environmental. Okay. So this is quite common in some population studies. So this came from a study in European individuals, and what you can see here, which is what I told you earlier, is that sometimes individuals that they come from different countries in Europe, which we look very different, you know, in a way. Maybe not to you, but when we are here, they are different. In fact, the genome composition is very different, but in groups. And this is a GWAS study in humans looking for some diseases, and what you can see is that I am Spanish, I am in that group. So most of the diseases that I have, they are shared with the Portuguese, which are my neighbors. And then what you find is you have many in Central Europe, that they share a lot of diseases, and then you have in Eastern Europe and even in Southern Europe. So basically, all those diseases are associated with markers, and we share markers in the population. And then this is the best example that these kind of GWAS studies can be very powerful in identifying markers of phenotypes in human populations or in any population. So in humans, they have been looking for ways to overcome the problem that I told you earlier, that is to have a very uniform genetic information. Like, let's go back to Indonesian population, very similar in terms of Indonesia, but you cannot identify enough markers to find some genes in disease, right? So one strategy that is being developed is this called administrative mapping. So what it means is that using a population that it comes from a location that it has a particular phenotype, and then using another population that is completely different. So for instance, if we use a GWAS study, then you take 500 individuals from Indonesia, but then you take 500 individuals from the US. And because you guys don't share too many markers, it becomes really strong the statistics to be able to connect one marker with the other. So most likely, most of the health organizations now, they use a panel of over 500 individuals that they are spread from US, Europe, Asia, and in some cases Africa. So this allows to create a lot of power in the statistics to be able to find those genes that otherwise are very difficult to find. Right, so this is for in humans. So in humans is very difficult because you cannot decide who is the mother, who is the father, who is the child. You know, this is done randomly, right? But in other systems, like things that we domesticate, animals, plants, we can control the parameters. We can control the population that we are going to study. Okay? And if we use plants, the way we do it is that we select 2 plants because we know that they are genetically very distant. Okay, that is the first piece of information. Then we generate an F2, and then from there we generate an F3. And then we separate all the plants that they have. They are sweet on one side, and all the other plants that are bitter on the other side. We extract the DNA from all these plants, use the markers in the genome to tell us what is the genetic composition of one of them, make that matrix, okay, and then basically run the statistics. And then the statistics will send you to some regions that you have frequency, high frequency for a particular group of markers, okay? And this you can actually see if you have the markers have a normal distribution in the population, so basically it tells you if you are right or not. And then in a way you can narrow it down to the gene. You can just go to the region and say, okay, this is the gene region area, so that one it has a particular mutation there or a particular change. So this is how we traditionally use QTLs in plants. Okay? So it's the common strategy. And this is why we use to improve plants. There is another strategy that we use in plants, which is, this is an example from maize. So you take a plant, you don't cross it with anything, okay? But then you grow individual plants from there, and these plants produce individual plants, and so on and so on. So this is done usually in plants that they have a lot of genetic diversity. All these, we want to improve these plants, right? So, and then at the end, we end up with groups of plants that they came from one family. As you can see here, they come from family. Usually we test in the order of 2,000 families. It's quite 2,000 families. Then you get the phenotypes of all these families, and then not only one piece of information. Maybe you know how many seeds it makes, how much sugar the seed has, how early the plants flower, how long are the roots. I mean, you can go for a very long list of phenotypes, and then a long list of markers. And then you can put them together, okay? And you can put these plants even in different environments. So what this represents is the plants growing in 4 or 5 different geographical locations. This is because plants respond to the environment a lot. So you can put them in Kalimantan, you can put them in Malaysia, you can put them in Java, you can put them in Thailand, right? And then you get the phenotypes in all these locations. Genetically, they will be identical. And then you run the statistical test, markers for all them, and then you run the statistical test, and then you ask the question, okay, which gene controls root length? And I said, that is the one, chromosome 5, there it is. Where is the gene that controls flowering time? Oh, then you go to the right, there are 2 genes, chromosome 1, and so on and so on. So in that way, that is how companies, biotech companies, improve all the crops and all the plants that we eat, right? They just look for that type of information to select all the genes that they are responsible for all these phenotypes. So if you ever work for a company that does that, you will be doing either 3 things. Collect data, phenotypic data in the field, right, which is an important part of information, or collect molecular information from markers, DNA, or statistics. So these are the 3 things that the plant biotech companies need from you. Okay? So everything that I told you is about information that is in the DNA. Okay, this is the bad part. Not all the information is in the DNA. And we know about that, okay, because the DNA is in the nucleus, and the DNA is folded into chromatin by histones. The DNA can be chemically modified, and the way that these things work in the genome is important for phenotypes. So at the moment, we don't have a way to incorporate this data in the models. It's too expensive. Not even in medicine. We cannot use this in medicine yet. But I think in the next 10 to 20 years, we will be able to get that information as well in the data. Okay? And this explains all these diseases that you cannot find in history, because this is where the information is. Right, so as I told you, you work for a company, either medical or plant, biotech, you will be doing any of these things, and this is how it looks like now, the work in these companies. So you work with a plant that usually is cultivated, most likely nobody has sequenced the genome. Let's say for Turkey, right? So still working with black rice, which is quite valuable in Indonesia, but nobody has sequenced the genome. The only genome that is available is the Japanese rice or the Indian rice. And this is not helpful because it doesn't tell you all the information. Okay, and this is very common all over the world. Africa, South America, most of the plants that we grow locally, nobody knows anything about them. Okay? But this is the same as well for human diseases. Right? There are many populations in the world that we don't know anything about, like Africa, maybe some parts in Southeast Asia, maybe South America. We know quite a lot about US and probably Europe, but apart from that, not much. So that means you have to start with a new genome, and that means you need genomic data, and then you need phenotypic data. So if you are working the genomic data, which is one of the jobs that you would have, is that you extract DNA, and then you sequence the genome. And for that, then you need to put the pieces together, assemble them, and then annotate the genes. So that is relatively simple, right? There are tools, people can train you how to do it, it's not bad. But the other part is the phenotypic data. So if you're working with plants, animals, then you have to collect phenotypic data. So you are working with plants, all these things that I told you earlier, but if you work with chickens, maybe you are looking at different phenotypes, completely different. Or fish, right? So it's a different phenotype. And for that, it's much more time consuming. Because imagine yourself going to the rice field to collect data on grain number, type of the plant. It's a pain, right? It's hot, it's sunny, it's very slow, it takes you forever. So companies are developing ways to be able to get phenotypic data much faster. Okay? And I'll tell you about this later. So there's a lot of development on that part. And then the important thing that we all want in humans or in plants is to get a lot of phenotypic data, as much as possible. Because the power of these studies is not to look at one phenotype. The power of this study is to look at 1,000 phenotypes with all the genomic information. That is the powerful part. Okay? And then once you get that, then you can annotate all the phenotypes, and then the last part is to integrate the two things together with the software that I told you earlier. Okay? So in fact, the analysis is easy. The complicated part is to get the genetic information and the phenotype information. That is the part. Okay, how can you do this, right? What are the ways to do this? So there are many ways, but I told you one way. So this is like the old-fashioned way. Okay, you don't go to the field, okay, which is good. So you can grow plants in a controlled environment. This is a glass house, a broad room, or you can go to a glass house, okay. And if you were working with animals, you will have something similar, farm, control farm, the control food, okay. And then what I'm going to tell you is that you can go to much more complex, because these ones allow you to grow plants in different environments or animals, okay? But it's a limit because it's expensive, right? You have to use cooling, you have to use light, and this is electricity, and this costs a lot of money. And the other option is to grow plants in the fields, outside, which is much cheaper. And then you have all the environments, so you can have a field here in Java, one in Kalimantan, another one in Thailand, another one in Philippines, and then this gives you all the environments for the region, and then it's better. Right? So this will allow you to look at phenotypes. But it still doesn't help you with the phenotypes. The only that helps you is to create environments. Okay? Okay. So the way is to automate the collection of information. So you don't need to have a human. Okay? So if you work with plants, and in humans as well, and some animals, then one is taking images. pictures, right? And as you know from Facebook, right? When you, or I have an iPhone, so you have an iPhone, take a picture and then immediately recognize all the faces, right? And it tells you who they are, right? So that is what this is doing. It's taking all the data they can take from the images. And this is all the possible data you can get from images and more, right? So the second possibility is that you don't take a normal color image, but you can use an infrared image. And if you take an infrared image, what you get is water content, or even the soil. So this is another information that is quick. And if you take a far red imaging, you can take temperature of the leaf, you can take salt, you can take shading, a lot of information you can take from images. And then there is a bit more advanced tools, like looking for fluorescence in the chlorophyll, and this tells you how efficient your plants are in producing carbon from the light. Or you can use hyperspectral imaging, which it tells you about carbohydrates, pigments, this is for projecting, and proteins, right? So it tells you about all that information. And then you can take carbon isotope measurements, far red, infrared imaging, and this is for cellular localization of metabolites. This is a much more focused, no large scale, smaller scale kind of measurement. And probably there are more than this, but I just came up with all these because we use this regularly. So it's a lot of data, and this, it doesn't require people to go to the field, right? But it has to be done in a glass house or in a controlled environment. That's the problem. And this is how they look like. So you can have this fluorocloud scan, where you can have plants here, and then you have a camera that is just going scanning through the plants, and then just taking all the data for the images. Or you can have these tri scans that when you see produce a blue light, and then this reflects the infrared, so you can take images of that. Or you can have special cabinets where you can have combinations of normal light, you know, in color as well as infrared, and then you can measure all the parameters constantly. Okay. Again, it's a huge amount of data that is collected there. I mean, think about the pictures you take in your phone. How many megapixels? How much memory you need in your phone? Because you're always running out of memory. So this takes basically pictures every second or every minute. So this creates a huge, huge amount of data. Okay? And then if you take that, you can measure morphology of the plants, photosynthetic performance. If you take this other type, you can take other ways to look at temperature, many parameters basically. And this is how they look like. So when you grow plants that they have a different phenotype, like for instance here, you can look up group of plants here that they're not blue. So this, what it tells you is that the respiration capacity of all these plants is very different. Okay? Or you can take the infrared and then look at metabolic variation, and what you can see here in red is some sugar concentrations, and then you see some that they are so red that they even glow outside the soil. So blue is the soil, and these are glowing so much, they're producing so much metabolites that they produce a lot of infrared spectra. So in this way you can have thousands and thousands of plants going through your screen, and you are collecting the data without having to touch anything. It's done with cameras. This is good, but you have to have all these plants in a controlled environment, and it's expensive. There is another way, which is still within the controlled environment, but growing them in glass houses. So this is a large scale facility. This is used by many biotech companies now. This is called an LiveStream platform. It looks like this. This is usually connected to a glass house, okay, where you grow your rice or your maize, and then there's a robot that it takes your plant in the pot, put it in the conveyor belt here. Well, you don't see it, but it will continue, and then just move it forward, rotate the plant here in this chamber, and they take images of it. And it does this continuously, day and night. And then with this, you can measure the growth, you can measure everything, right? And for animals, they have something relatively similar. So I have seen one that they have for cows, and basically cows just walk past, and there is a digital camera that takes measures of everything, make volume, the skin thickness, check the weight. So different companies use different strategies, but it's all computerized to collect data. And then like that. So you have a picture, then you have to render this image into pixels with different values, and then you integrate this data into the dimensional view, and then from this you can get a lot of data. And so it's a way to computerize, make data from images and from any other value to help you to understand the phenotypes of these large populations, right? So this is one of the major sources of income in Southeast Asia. So you have coconuts and trees. You cannot grow them in glass houses. You have to grow them outdoors. You could grow them in houses, but it would be an expensive thing. So many companies in this part of the world, and in South America, and in Europe, right? So it's, in the field, you can take data. So you have like a little buggy that goes around with the camera, and you can collect all the data. Okay, and you can do this for millions of trees. Okay, but this is not enough, because we are talking millions of hectares. So to get information to millions of hectares, you have to look at this. And this basically is a satellite image that it tells you all these parameters that I told you earlier. Okay, and from this data, you can extrapolate pretty much everything that I told you earlier. You can extrapolate the height of the plant, you can extrapolate the photosynthetic value, resistance to drought, everything. I can tell you that the amount of data is so large, okay, that there are some well-known companies like IBM, which has invested a lot of money in the analysis of satellite data for many different companies. And this is not only for plants, because IBM has invested a huge amount of money in medical phenotypic data. So they are in every hospital pretty much in the world collecting data. Every time that you go to a hospital and then you have a minor thing, then all that data gets recorded and then put in a database, and IBM and other companies, they are actually analyzing the data in the same way that Facebook is looking at your habits and then making these pop-ups with all the things that you like, right? Like you are thinking to travel to, Singapore. Then for a week, continue popping, Singapore flights and hotels in Singapore. So this is the same thing. It's just using a lot of data to be able to predict phenotypes. And this allows all these companies to get phenotypic data in the natural environments. And this is much more powerful than anything that we had before. So this is a field in the UK where different plots of wheat have been grown, and then basically this is an infrared, this is a drone data, okay, on the different plots, and then from here we can extrapolate different values from the different phenotypes of the plants. The advances are so massive that there is no a single rule of how to collect data. So if you look at plants that we grow, there are terrestrial data collector. So this basically is this came from a company in the UK. It's a mini robot that actually runs through a plantation of lettuce, right? And it can run. It maybe has millions of lettuce plants and run through it. And it has a little sort of aerial here. It has a camera and it has sensors, and basically it's constantly recording data in a particular path of these millions of lettuces growing. And from there, it collects all the data, the composition of the soil, the temperature, the growth of the plant, and it is very valuable because all this data goes to a computer server, and then a company will analyze the data, take phenotypic information, which you can use for GWAS, but as a farmer, you might want to know if your plants are getting infected by a virus or by a pathogen. And this device will tell you automatically that this is happening in your field and exactly where. So you need to treat these plants, you will precisely know where to go. Okay? And this is another, for those of you that like technology and engineering, this is a big area of interest where to go. And of course, you have drones, like here. In Australia, there's a lot, in Malaysia as well, and probably here in Kalimantan as well, there's a lot of plane spectral imaging, so to collect data with planes. They just fly and collect data. And then, of course, a lot of satellite imaging. This is for very large plantation, in the US, all the corn fields data is all done with satellite imaging. There are even for animals as well, where they count the reproductive success of cattle in Argentina as well. In different farms, they can actually do that as well. So it's collecting phenotypic data, in a way, using different technologies. Right, so let's go back to the technology. So this tells you, how you can go from statistics, markets, how you can collect markets, you can do universal studies. But there are other ways to inform you this, okay? And I want to give you a couple of examples, 3 examples. Okay, one of them is that when you discover something in one species, it can inform you about what is happening in another species. So let's say, for humans, okay? So if we discover a gene that is important for hyperglycemia in mouse, this could be very useful because then you can translate this into humans. And this is why, for many years, biologists have been using model organisms. You can think about Drosophila to understand neuronal development. You can think about mice to look at health, aging, some diseases. You can think about C. elegans, the worm, to look at aging as well, and some diseases, and you can go on like that. So there are many, frogs for looking at reproduction. So you could take that information into humans, then it will be really advantageous, right? And because now we have sequenced genomes, now we can do that. And this field is the comparative genomics field, because now everything we do in plants or in animals, and if we want to understand phenotypes, it has a component where we are using information from other organisms. Okay? So what this picture represents here is the 22 chromosomes plus the sexual chromosomes of humans, and in blocks it tells you all the conservation that you have with mice. And you see 2 things. There is a lot of conservation between human and mice, but the location of the genes in the chromosomes, they are not in the same chromosome. Okay? So let's say one example. One example that is easy to see because there are many colors. X, they're all in the X. So it's good. Sexual chromosomes, very good. Not everywhere, but But then let's look at one that is easy to see. I don't know which one, the red color. Chromosome 2, okay? And then in mouse, and then you see a part in chromosome 2, another part in chromosome 10, 11, and so on, right? So it's all fragmented, basically, right? And why is fragmented? Well, the reason is because all these genes undergo a gene duplication at some point. So humans, we have a duplicated genome, like most eukaryotes, okay? And then this has created usually 2 genes, okay? And then these 2 genes in the 2 species could be in different locations. So they can be located in different places. But the genes, the function of the genes may persist, okay? So many genes evolve with duplication of all ones. So if we have an ancestral gene that we know in a basal organism, most likely we will know in species number 2 where it's located and how it works. Okay? And then you have 2 different types of genes, the orthologs, which are genes that are separated by species events, so they have very similar function. But then you have paralleles that they are done by duplication, and these usually the genes have different functions. So you have a parallel, is very similar gene, but it has a different function. Okay? And knowing what is a homolog and an ortholog and a parallel is very useful for comparative genomics. Okay? So then, if you compare 2 genomes, one on the top and one at the bottom, then what you can see is that you can have a lot of rearrangements in the genomes. But if you have all the markers, if you know the sequence of both genomes, you can predict all the combinations that you have in all the chromosomes. So the information is scattered all over the place, but you have the power to be able to know where different things are. Okay? And if you are able to get this information from the duplications, then you can build synteny maps. So this is, as we call it a circus plot. But basically what it means is a circular representation of the chromosomes in the genome of different species. Here in red, you have rice, right? Next to it, you have foxtail, and then sugar to each other. Okay? So if you are trying to improve, let's say, maize, which here in the outer part, then the information that you have from rice is incredibly powerful, because whatever gene you find in rice, most likely is going to be important in maize, and the other way around. The maize gene that you find with a particular phenotype will send you the same gene in rice. So quickly, you can connect the genotype and the phenotype without having to do all these GWAS studies. So speed up your work. And what you see is that sometimes the synteny is so big, like for instance, this is the waxy. So this is the gene that is connected with the content of the starch in the rice grain. And then you can go and rice, it's located in this chromosome. It has all these other markers nearby. And then in rice, in maize, is in another chromosome. But then it has very similar markers that they are conserved. And then the location is not the same, but it's in the same interval. And then you can go to rice. There's another gene that is here, which was in fact discovered in maize, which is for architecture of the plant. And then you find the homologue in the other chromosome in rice. So you can go in either direction from one genome to the other to understand the phenotype and what are the genetic bases of the phenotype. And this is what companies do all the time now. So if you are doing discovery of traits, then you are not only doing the GWAS and all this stuff, but you have a team that they are actually looking at comparative data. And this you can do it like this, but there is another way to do it, which is based on Google research, which is looking for text information. Because now we have so many publications in many scientific articles that if you're going to say, if you ask in Google, starch content, rice, and gene, Google most likely with the algorithms they have, they will send you to that gene. Just by looking at all the information that is in the web. So you can use that word information to even inform you of the connection between the genotype and the gene. It's crazy, but you know, it's the way that these companies have developed. So we are using this as well. So you have synteny, you can use that information, but there's a small complication here. There's a small complication, and this is a big complication in plants. And what is highlighted here is evolution in plants. So let's start from basal plants, okay? Unicellular, let's say algae, right? The little algae that grows around. And then you go here to rice, right? And what you see here is that when you look at the content of DNA, it has increased very rapidly from the single algae to the basal plants to the more complex plants. So in fact, most organisms have created a genome that is bigger and bigger and bigger. So that means that it's more difficult to create synthenies. And actually you can see it here, because in this slide what you see is that maize, it has two circles, right? You can see here. And the reason why it has two circles is that maize is an ancestral polyglot. It has duplicated the genome, right? And the other thing that you can see here is that rice, it has a very small red little bit, but this is getting bigger, bigger, and bigger. So the genomes of some plants have become bigger and bigger and bigger. And I call this genome obesity. So they are getting fatter and fatter, right? And the question is, why plant genomes get fatter and fatter, okay? Well, we don't know. The answer, the quick answer is we don't know. But there is something interesting. So if we look at one very small genome, like Arabidopsis, then this has small cells, it has a fast reproduction, and it's very good for habits like weeds that they spread everywhere. But if you find plants with really fat genomes, like this Fritillaria, which grows in alpine part of Europe, or Paris, that grows in alpine part of Japan, these are huge. I mean, this is like, you know, 1,000 times an Arabidopsis genome. This is 500 times the human genome, right? So this is 5 times 40,000 genes. These are huge. And all of these, they have very large cells. They have a slow reproduction. And this one is very good to make bulbs, like peanuts, right? Well, something below ground. And then this one is very good to grow in response to freezing. So it can tolerate completely covered with the snow for several months. So clearly evolution has selected plants to get bigger and bigger genomes. And I'm afraid that the crops that we use, we have domesticated them to get bigger and bigger genomes. So here we eat a lot of melons, you know, watermelons. Yeah. So watermelons have a huge genome and polyploid. So las companies that try to improve melon have a lot of difficulties because they are dealing with these huge amount of genes, right? To interpret the data, to connect the genes to the phenotypes. So bigger genomes are complicated. And genome obesity, you can find it in archaeological samples. So this is an example of a cotton that it has taken from Egypt in the pyramids, okay? And it has taken from South America, where cotton is grown as a cultivated in the last 500 years. And this represents in blue the amount of genome in the archaeological to the current sample. And what you can see here is that it's becoming really huge. It's pretty much 4 times the genome size of the original cotton, almost 5,000 to 10,000 years ago. So in 10,000 years, a plant that we have selected to grow and make clothes, cotton, then it has increased the genome massively. So it's complicated to connect genes to phenotypes when this happens, right? So my last example is connected to this point. So why archaeological record could be good, right? And actually yesterday I was with Yekti in an archaeological site very close to Yogyakarta, where they have, you know, samples of these ancient Java man. So actually, archaeological record now is becoming a very good tool to connect the genotype and the phenotype. And actually, it's weird, but it is. And there are probably over 10 to 20 studies in the last couple of years explaining this. So this is a study published in Nature from this Davisonian discovery in Siberia. This is a sample of the only of the teeth that it was collected from. You can find some of this as well in this archaeological center. We saw some of them. From this, in this case, they were living in caves. They extracted DNA from here. They sequenced the genome. Okay, and this is where Homo florensis from Indonesia is located. So it's a very ancient human species, and this one is very close to the Neanderthals before the Homo erectus actually evolved here in Indonesia and Europe. And by having all the genome information of this, then we can look for association between phenotypes, diseases, and genes. And this is one study that they did in this work. So they compared genome information markers in Homo sapiens. Now they have one sample of the Neanderthals. And then what you can see is that there is one region here that is absent in the chromosome 2 of humans, current humans, which was present in the Neanderthals. And this region, so that it was absent in the Neanderthals, and actually this region is for a lactose intolerance locus. So the early humans basically lost that gene, okay, to be able to use milk, consume milk after birth. Because most animals do not consume milk after birth. They only consume it from humans. But humans, we can do it for a long period of time. Okay? And that is the point that links one gene to that phenotype, and you can actually go back in history 10,000 years, and you can find that that is the region of the genome that explains the phenotype. So you can actually go and give us studies in plants, in humans, and this is probably why you will see on the news a lot of things about archaeological samples. You know, archaeological samples here in Indonesia or in China. There's a lot of interest in archaeology in China to be able to obtain this kind of information. Right. A long lecture. Sorry. But I think what I want to tell you is that if you want to use this, you work in a company or you are involved in a medical study, or you ever come across with this, these kind of studies are big data studies, right? So these are connecting phenotypic information and genotype information, because now we can, right? Something that we couldn't do 10 years ago. This requires a lot of statistical and methodological challenges, like, you know, having the right samples, having the right markers, being able to determine the phenotypes quickly and cheaply. So it's a lot of things on there. But the phenotypes is the biggest challenge. Surprisingly, for a biologist, the molecular biology component is easy. You know, it's expensive, but it's easy. It can be done. But the complex part now is the phenotypic part, which initially everyone assumed that it was going to be an easy thing, right? So that is going to be the largest limitation to this. And this is why everyone is looking at ways to get around it. And the only way to get around it is artificial intelligence, which what it means is to use computers to learn from patterns or learn from images, like you know, iPhones, Facebook, and all these companies do. They can learn from that and then create that into numbers, and then use computers again to analyze the data. So I am afraid that there is not too much use for biologists looking at the microscope, but there is a lot of use of biologists to interpret the data and to design the experiments, because this is what is really important. When this will be cracked in probably 5 or 10 years. Well, probably it's being cracked already, right? And we don't know. Well, I can tell you it's cracked already, but you know, this will be, you know, it will become mainstream, let's say, you know, in 5 to 10 years, right? Then when this is done, then the important thing is about planning experiments, you know, managing population, collecting data, interpreting data, all this. That is right. I just want to thank you. I'll put my email address here in case you want to get in touch with me, and Yakti will have a copy of my presentation. Thank you.
Thank you very much for proposing for a valuable and interesting talk. Now we are heading to question and answer session. Please mention your name and your evaluation which lab. And yeah, any questions? Yeah, please.
My name is Hasil from Biotech Lab. I want to ask, in several weeks ago, in Science was published research by Broad Institute that revealed same-sex behavior influenced by not only one single gene but many genes, and used from UK biobanks data, and the research was used US methods. How accurately and effectively the methods to the future of genetic disease research and genetic threats research. Thank you.
Yeah, thank you for that. So in fact, that project has been running for almost 10 years. So the UK has decided to sequence the genome of 1,000 to 5,000 different individuals from different ethnicities. So actually in London, it's quite lucky because it has a huge Asian population, it has a huge European population, it has a huge African population, it has a huge Central American population. So all these are migrants, right? And the problem that I told you of having genetic structure is not a problem in London. In fact, in London, there's a huge diversity of genomes and phenotypes. And all these people go to the hospital, and all the data has been recorded for the last probably 40 or 50 years. So now they have a huge amount of power to understand many diseases, and this is one of them. They have published several papers, but yeah, incredibly powerful. The previous model system to study this kind of disease, it was Iceland in in the north. But this, it was very good because it had a lot of medical records for a long time, and they sequenced the genomes of many people from Iceland. But the problem is that they were all very similar. And by being very similar, they are not good for most diseases. So actually this was a very lucky or clever decision of choosing that part of UK, because Wendy can tell you that London is very diverse, but when you go to other cities far away from London, you find less mix of population, right? So you have different ethnicities, probably some people from India, but not many from Southeast Asia. So London is a perfect location for that experiment. So yeah, I expect, and in fact, all the data is public. So I had 3 students last year in collaboration with another researcher that was looking at the data from the biobank. Anyone can go and collect the data from the biobank and say, okay, I'm interested in this disease. And then you can run your own GWAS to find locations. So the data is publicly available for everyone.
Thank you, Sahar. Is it answering your question?
Yes.
Oh, thank you. Yes, please mention your name and your laboratory.
Thank you for the chance. My name is Anissa. I'm from Genetics and Breeding Laboratory. My question is simple. Like, genome obesity or genome get bigger and bigger, is it related like in surface gene theory or surface gene DNA in Richard Dawkins' book?
Yeah, it's exactly that.
Oh.
Yeah, that's a good point.
Okay, thank you.
Not only that, but yeah, this is one of the factors. So there are 2 factors. One is in plants in particular, is polyploidy, which means duplication of chromosomes. So that happens quite commonly. And most of the plants that we eat, in fact, have 2 or 3 times the number of chromosomes compared to the wild species, because for 5,000 or 10,000 years, humans have been going for the bigger fruit.
Yes.
And more chromosomes means bigger size.
By the way, that's,
But in terms of evolution, right, across different species, the increase, it has been in the repetitive part of the genome, right, in these selfish genes, right?
by the way, that's also the interest in my life.
Yeah.
Okay, thank you.
Yeah, good point.
Thank you, Lisa. Before going to another question, we have, because it's now broadcasting in YouTube, and someone has asked the question. So this is from one of our lecture, actually. He's working in biochemistry lab. She says, currently I'm working with CIRAT on rubber. CIRAT has the full genome on rubber already, and we are also working on the response of rubber clones to drug. Is there any limitations number of clones or replicates? I mean, maybe in this GWAS analysis.
Yes. Well, It depends on the genetic diversity of the rubber-free collection. So if it's being propagated as a clonal, then the genetic diversity will be low. So increasing the number will not help. So for GWAS, the most important thing is to have genetic diversity. So, and large numbers.
Okay, thank you. Well, but continue this question. Actually, is it any related with what we call it technical replication and biological replication? Because if say that if a species is really hard to get the replication, let's say that you have transgenic plants that you have to work with small number of replication, and then is it okay with having
replication will not help in this particular case to the statistics. It's about the number of variants that you have.
So there is any, what number, like numbers? Can you mention a number?
Depends on the size of the genome and depends on the number of markers in the genome. For humans, it's in the order of 10,000.
10,000?
Yeah, markers. And then maybe the smallest GWAS study could be 2,000 individuals, 1,000 individuals. More individuals is more power in the statistics, yeah, and more variation.
And it's better if it's propagated sexually, not clonally.
Sexually will be better because you get maintained variation in the populations, okay, which is a problem for rubber and for other plants propagated clonally.
Okay, thank you, Jose. Is there any question? Yes, please.
Okay, my name is Muhammad Fadienda from graduate study of biology. I want to ask, from your examples, it's all about bigger organisms like plants, animals, and humans. But in microorganisms, they have a low genetic diversity because most of them is reproduced asexually. So how do we get the genetic diversity from the microorganism? Thank you.
Yeah, I think in microorganisms you have another tool that is as powerful, which is the capacity of targeted mutations. So you can engineer targeted mutations and you can get to the phenotype in that way. This is being traditionally used for yeast, even for E. coli, just to generate random mutations and then select for phenotypes.
So we still use the GWAS for that?
I don't know of too many or any study of GWAS in microbial systems.
But is it possible to use?
I think it's possible. You have genetic diversity and capacity to look for phenotypes. But because of so powerful, the capacity of creating mutations so powerful in microbes, then there are other ways to get to that information.
Thank you, Mohammed. Thank you, Jose. Next question. Yes, please. Yes, please.
My name is Vicky, and I have a question. I just like the same with this question, but about the macro genetic diversity, how to study about the how to improve the medicine about to cure some disease, maybe like undetectable virus like HIV and like this provoke macroorganism in the medicine with the GMOs. Can we solve that problem with these GMOs?
Yeah, I'm not sure if the sample that you gave us it could be used with GWAS. Certainly, looking for response to infection. You know, there's been some studies of GWAS in humans, but not for treatment. Right? This is basically to identify groups of individuals that they are at risk to have a particular disease, you know, viral or bacterial. I think that is more powerful in that sense. If you think about treatment, you know, I think, you know, you always have the immune system as the best way to activate. So vaccination, you know, there's a recent development in Ebola where is a vaccine is going to be deployed. Maybe for HIV has been several attempts to create vaccines to control viruses. So I think for this kind of infectious diseases, it's not as powerful, this information.
Thank you. Next question.
Talking about the ancient DNA.
Mm-hmm.
This maybe is also sometimes come out in my mind about the we have a lot of situs in Indonesia that maybe the archaeologists also working in that. But how to that connected between the old generations. That the old I mean that we have a lot of things that maybe animal or human also plant, and connected with now we have a diversity of the plant, maybe domesticated one, and maybe the ancient in the old time, maybe also we can also doing something like a was analysis for doing they connected between the old time and also the modern time.
Yes, certainly. One of my colleagues, Robin Alabi in Warwick, has been leading research on the area, and he's been looking, some of the data is what I saw you, and he's been looking at maize, archaeological samples, cotton, barley, many different species from archaeological samples. And this is incredibly valuable, to understand domestication, the genes that are important for domestication. So I think it's anything that it can help is good. And I think this is a source of information that it can be used.
So how to isolate the DNA from the ancient? The ancient DNA is also.
They have established protocols for that. Yeah, it has to be done in special labs to not to have contaminations. And the sequencing, it can be done.
Yeah.
The DNA is usually small, fragmented, but it can be assembled.
Yes. Next question. I have a question. Is there any chance that if you collect samples, it's from the same, like it's same sample, let's say it's technical sample, like it's say from one ancestor, let's say there is no, it's diploid, it's not polyploid species. Is there any chance that you can get different results in your GWAS? Like let's say that it's not grouped together in a PCA, or it's different, but actually it shows different grouping, let's say. Is there any chance, or if that is actually happen, what is the cause of it?
Oh, okay. What is the reason of that? Usually it is a small sample number. So when the sample is too small, then you can have any of these effects that affects the the structure of your markers are stronger. And this is why now it's thought that to be able to have a good solid GWAS, you need to have a very large sample, especially when you want to get close to to the gene of interest. You are only interested in a region of the chromosome, maybe, right? But sample number is fundamental. You need to have a big sample because you need to have a lot of data that is sending you in different directions statistically to get rid of them.
Well, thank you. Next question. If there is no question anymore, then you can, if you have doubt in your heart but you don't want to ask, you still can get in touch with Jose in his email. Well, I think that's the end of our Q&A session. I would like to say thanks again for Jose for a valuable, interesting, and lecture for us today, and also for our audience for your active participation. And hopefully this lecture will be beneficial for everybody Thank you very much. Good afternoon and wassalamualaikum warahmatullahi wabarakatuh. Okay, before we leave this room, I think we can take a picture together with Jose. Oh, I'm sorry. In this time, we would like to give listen for Jose from Faculty of Biology. I will give this to Buyati. Okay, I would like to invite us to stand in front and we can take some pictures, group picture together.